So with this, continuing with the good science which you've been listening all through for the last 3 days now, and some of the eminent, excellent speakers. And in this session as well, we have to start with the first speaker, Bob Sharp, who will be talking on physiological control of maize nodal root growth under drought, a re-heightened priority. So you will see that how important it is, as he's emphasizing the point, re-heightened priority, and we look forward to listening to his talk. And I request Bob Sharp to kindly make the presentation. Bob Sharp, the floor is yours.
Thank you very much, and good morning, everybody. So what I'm going to try and do over the next 30 minutes is tell a sort of a story that starts with my original studies back when I was a graduate student, focusing on maize nodal root response to drought, then taking quite a lengthy detour looking at the primary root of maize seedlings, and then back to our more recent current studies, refocusing on the nodal roots, hence the title, A Re-Heightened Priority. We've heard a lot in this meeting so far about the importance of root systems as a critical component of plant adaptation to drought. The physiology of root growth responses to drought, however, still remains fairly poorly understood. This is, of course, partly because roots are underground and experimentally difficult to study. It's also because root systems, as we've heard in the conference, are very complex. Different types of roots are experiencing different conditions at different places in the soil profile and at different times during this duration of drought. So this overall complexity that you can see in this classic diagram from 1926, showing the overall plasticity of root system development in drying soil, encompasses a huge range of different responses. I'd like to focus on one specific aspect of root growth response to drought, and that is the ability, in fact, the necessity of certain types of roots to be able to grow through dry soil to find moisture at depth. And this is a characteristic of the nodal or crown roots in grasses, such as maize, and also of the primary root of developing seedlings. Now, maize nodal roots that we've heard a little about already in the conference, just to remind you, grow from the base of the stem from a successive series of stem nodes. And because of that morphology, in a drought situation, there may always be some young nodal roots that are being produced directly into soil, surface soil, that may be extremely dry. And they have to be able to grow through that soil to be able to find moisture at depth. And it's critical that they do so because the nodal roots produce the framework of the mature root system and can penetrate down a couple of meters or more into the soil. In this situation, and I'll come back to this later in the talk, the direction of water transport is the opposite to what you normally think of. The root that's growing through very dry soil is not taking up its water directly from the soil. It's actually deeper roots are taking up the water that is both being used for transpiration and also is moving downward through these roots to the root tip. That point is explicitly made when you consider the older generations of nodal roots that obviously have to grow through the air first before they penetrate into the soil. So clearly getting their water from the base of the stem. Now in that sense, one can sort of make an analogy with the growth of leaves, which of course also grow typically into a very dry aerial environment. And in that sense, this is what these roots are doing as they penetrate into very dry soil. So although what I'm going to talk about today specifically addresses mechanisms of root growth regulation, I think the principles also apply to the growth of the shoot as well. Perhaps it's no surprise that it was shown in earlier work that nodal roots have a very high capacity to maintain root elongation at low water potentials. This is shown in my early work. As I said, I started my PhD on this topic, and this is my very first paper, Sharp and Davis, 1979, and also a classic study from Mark Westgate and John Boyer in 1985. And both of these studies showed what's shown here from Boyer's paper, that nodal roots have the ability to maintain elongation at lower water potentials than those that inhibit shoot growth. Both of these were studies of osmotic adjustment, and really this remains the only information that we have on how maize nodal roots grow at low water potentials. There's been no further information in the literature on the underlying physiological mechanisms of maintenance of nodal root elongation. So I'm going to return to that later in the talk and take a little bit of a detour and briefly talk about our work on the other system in which is maintained at low water potentials, and that's the primary route of developing seedlings, again in maize in this case. And the reason we switched many years ago to working on the seedlings is because it's a much more experimentally amenable system. This is a well-characterized vermiculite culture system where the plants are grown under non-transparent conditions and are transplanted into specific calibrated water potentials. And under these conditions, the seedlings exhibit uniform, reproducible, steady responses. As illustrated here for a couple of genotypes, showing contrasting abilities to maintain primary root elongation under severe stress conditions. It's from a paper we published in Crop Science a few years ago. And this line, inbred line FR697, is a line that we selected for more detailed work that shows a superior ability to maintain primary root elongation, and as I'll show you later, nodal root elongation as well. Now, a key experimental approach that I began when I was a postdoc with Wendy Silk at the University of California, Davis, many years ago, published in this original paper in 1988, is the application of growth kinematics. Kinematics describes the spatial and temporal characterization of expansion patterns within an organ. And what we discovered in this original paper has been really fundamental to understanding underlying mechanisms of growth regulation. And that is that cell expansion is extremely differentially sensitive to water stress at different positions within the root growth zone. So cells are produced in the meristem and then are displaced away from the meristem by the production of new cells. And as they find themselves at different positions from the meristem in the well-bordered roots, they accelerate and then decelerate and stop at around 12 millimeters. What we showed in this 88 paper was that a remarkable ability to maintain cell expansion in the apical region, followed by a zone of premature deceleration and shortening of the growth zone. Now, we've actually focused most of our interest over the years on mechanisms of growth maintenance in the apical region. I'm not going to talk about that today. I refer you to this fairly comprehensive review we published in PCE in 2010. What I'd like to briefly consider is the question of why cell expansion is not maintained under stress in the basal region, and if we could understand that, we might be in a position to improve root elongation under stress. So I'd like to just briefly discuss some work on cell wall biochemistry related to the role of ferrolate cross-linking as a potential restraint to cell expansion in that basal region. Our interest in this arose because of a transcriptomic study that we published a few years ago in this system that suggested that enhanced synthesis of ferrolates may be occurring under water stress in this region. Ferrolates are phenolic compounds which are abundant, particularly in cell walls of monocot plants and particularly in grasses. And they are believed to play a role in cross-linking the polysaccharide matrix within the cell wall and thus restricting cell expansion. So we've been addressing this by a number of ways. I just want to show briefly some results using some ferrolate deficient mutants that we were fortunate to obtain from Ron Phillips at the University of Minnesota. Ron had isolated these mutant lines with an interest in cell wall digestibility related to forage and bioenergy reasons. Our interest was on growth regulation to test the hypothesis that increased ferrolate cross-linking may be an important limiting factor for cell expansion. In other words, that ferrolate deficient mutants might show enhanced ability for root elongation. These images, hopefully you can see at the back, using a modified autofluorescence technique with this green fluorescence indicative of overall cell wall bound ferrolate levels, shows that there is indeed evidence of a substantial increase in ferrolate levels in the basal region of the wild type under water stress, as was predicted from the transcriptome study. And that in the ferrolate deficient mutant, SFE mutant line, there's evidence of reduced ferrolate levels both in the well-bordered and particularly in the water-stressed root in these basal regions. And as we hypothesized, this ferrolate deficiency was indeed associated with an enhanced ability for root elongation under both conditions. This is the results from well-watered roots, and this is the cell length profile shown in the upper right as a function of distance from the tip. So the cells expand until they get to the end of the growth zone, fully expanded cells at around 12 millimeters. And in the wild type, at about 12 millimeters. And as you can see in the mutant, there's just a continuation of cell expansion, so the cells continue to expand and reach a significantly longer final cell length at a greater distance from the apex. So in this kinematic analysis, you can see that the length of the growth zone is enhanced, and this overall response leads to about a 12% increase in steady root elongation rate. And the same is true and more pronounced in the water-stressed roots, In the zone where there's normally premature growth deceleration under stress, you see a substantial enhanced ability for cell expansion occurring in this region due to a further ability to maintain cell expansion, leading to about a 20% increase in final cell length that was associated with a 29% increase in the overall root elongation rate. So that's exciting initial data. An obvious question is, does the effect of reduced ferrolate levels extend beyond the seedling stage and lead to enhanced root elongation at later stages? And specifically, I'd like to begin to reconsider now nodal root elongation. So to begin to address this question, we conducted 3-week soil drying treatments of plants grown in 60-centimeter tubes in a growth chamber and examined the root morphology at the end of the experiment. And as you can see, that certainly the SFE ferrolate deficient mutant does indeed show substantially enhanced root development compared to the wild type in these slightly older plants. So this is a substantial enhancement in root biomass and root elongation seen in the well-watered mutant line. And this is sustained under soil drying conditions. Now, what I emphasize, this isn't simply that whatever happened in the well-watered is just carried over to the water stress. In order to sustain this effect under water stress, as I've emphasized at the beginning of the presentation, the nodal roots must show an ability to maintain growth during as soil drying occurs, particularly in the upper part of the profile. And if we look at the nodal root lengths associated with this experiment, you can see that ferrolate deficiency indeed does allow an enhanced ability to maintain nodal root elongation. This is node 1 and node 2 of the wild type and the mutant under well-watered and soil drying conditions. And you can see at the time that node 1 was developing, the soil was, which is in a younger plant, the soil was not dry enough to cause any inhibition yet in the wild type, so no effect in the mutant. But by the time node 2 roots were emerging, a few days later, the upper soil was obviously dry enough to significantly inhibit root growth in the wild type, and that was completely prevented in the ferrolate deficient mutant. So these initial data indicate that ferrolate deficiency can lead to enhanced ability for root elongation under water stress, both in the primary root of seedlings and also in the nodal root of slightly more mature plants. Now, the other point I want to make from this slide is to begin to introduce the complexity of studying nodal roots in contrast to the seedling model system. And I've already made that point in describing this slide. At the stage that node 1 roots were growing, the soil and the surface was obviously still fairly moist and not dry enough to inhibit growth. But by the time node 2 roots were growing, the soil was obviously substantially drier. And this, of course, is expected in a progressive dry down experiment. But it makes it very complex then to compare different nodes of roots, because the different nodes are experiencing different degrees of soil drying. And also then it makes it very difficult to compare across genotypes. And so to tackle this question is part of a major new project that we have in the lab, which is really it's been funded recently by the NSF Plant Genome Program over the next few years, and involves a multidisciplinary team at the University of Missouri to really begin a comprehensive analysis of nodal root growth physiology under drought, moving from the lab using model system I'll describe in a moment, to the field, taking advantage of the field rainout shelters that were mentioned in an earlier talk this morning. So as I just mentioned, if you simply withhold water from the surface, then there would be a gradient in soil drying, and the different nodes of roots would be experiencing different conditions, both spatially within the soil profile and temporarily over time as the soil continues to dry. And as I've mentioned, that makes comparison among roots and reproducibility and comparison among genotypes complex. And so we've developed a divided root system approach to tackle this problem, which is really allows us to apply the same precision to study nodal root response to low water potentials that we've managed to achieve over the years in the primary root system. So in this system, the primary and seminal roots grow in an inner container, which we can maintain under well-watered conditions. And that water supplies both the water for transpiration, for shoot development, from which the nodal roots grow, but the nodal roots emerge into an outer container, which we effectively build around the inner container. And this outer container is filled with pre-calibrated, defined water potential media, as I said, by analogy with our seedling system. And so the nodal root experiences the same low water potential media as it explores through the soil, and subsequent generations of nodal roots experience that same condition. These are fairly lengthy tubes, as you can see, over 1 meter long, and that allows us to study nodes 1, 2, and 3 under controlled lab conditions. And I'd like to share some initial results obtained with this system. And this is comparing the responses of standard genotype B73 with that inbred line FR697 I mentioned earlier on, which was selected because it has a superior ability for primary root elongation. And as you can see here, that's also true for nodal root elongation. So I'll try to illustrate this. This is at the end of an experiment where the nodal roots have been growing through the outer container for about 7 days. And the root length of the well-watered roots in both genotypes is about the same, nearly 25 centimeters. And as you can see in B73, if we look at node 1 in blue, you reach a length of 10 centimeters with the soil only at 0.55 megapascals, whereas, I don't know what this is. Oh, you can't see that. Very strange. You reach a length of 10 centimeters in FR697 when the soil gets all the way down, only when the soil gets down to -1 megapascal. So there's a significant difference in sensitivity of nodal root elongation to a defined range of low soil water potentials between these two genotypes. However, note that this data is plotted against soil water potential. In the same experiment, if we plot the same data against the root tip water potential, the apical centimeter that encompasses the elongation zone, you get a different story. And now you can see the sensitivity of root elongation to low water potential of the tissue itself is actually quite similar in the two genotypes. B73 reaches a length of 10 centimeters at a water potential of the tissue of about 0.65 megapascals, and that is the same in FR697. The difference in the genotypes is the soil water potential at which that tissue water potential occurs. And you can see that if you look at the driest treatment here, or a soil water potential of 0.9 megapascals, then the node 1 roots of B73 actually have a root tip water potential drier than the soil that they're growing through. Whereas in contrast, at the same soil water potential, the root tips of FR697 have a considerably higher water potential than the soil they're growing through, and consequently have a greater maintenance of root elongation. Now, to re-express that data in a way that may be clearer for you, I've plotted the same data, but I'm plotting now the root tip water potential encompassing the growth zone against the soil water potential. And if the root tissue simply changed in water status as a reflection of the soil that it's growing through, you'd have this one-to-one relationship indicated by this dashed black line. And you can see that B73 is pretty close to that one-to-one relationship. In fact, it is in node 1 with a slope of -1 and a slightly higher slope, but still pretty close to that one-to-one relationship in node 2. In contrast, in FR697, a much shallower slope is obtained, indicating that FR697 has the ability to somehow prevent the root growth zone from experiencing the same low water potential as the soil that it's growing through. Now, we've extended this experiment substantially. This experiment stopped at a soil water potential of around 0.9 megapascals. And I'm going to add another treatment here where we drop the soil in the outer container all the way down to -2 megapascals. And that revealed a really quite striking result. That is, in FR697, the root tip water potential actually did not fall any further, despite a 1 megapascal drop in soil water potential. And that was true for node 1 and for node 2. This looks like it actually went up, but that's not a significant difference. And the same trend is seen in B73, but obviously to a much less extent. So these data indicate the induction of some form of mechanism or mechanisms that facilitates water transport to the root growth zone as the roots are growing through very dry soil, and particularly in FR697. The result of that is that root growth is very insensitive to that further reduction in soil water potential. So this is the same data plotted again, but with the addition of the root length at the end of the experiment. And as you can see in FR697 particularly, there's no further reduction in root growth all the way from -1 to -2 megapascal soil water potential. Now this data indicates some form of modification and difference in root hydraulic properties between the two genotypes. And these observations, again, took me right back to my original studies in my PhD, my early studies of nodal roots. And this simplified model actually comes from my PhD thesis in 1981. And I developed this as to try to explain some observations that Bill Davies and my supervisor and I made when we were studying osmotic adjustment in maize nodal roots. And that is that we showed in that original 79 paper evidence of steep water potential gradients that occur at the tip of nodal roots growing through drying soil. And we speculated here in my thesis that this was perhaps because a root growing through well-watered soil is likely to be able to take up water readily for growth across its entire surface, whereas a root that's growing through soil that's drier than itself, as I emphasized at the beginning of the presentation, is having to take up water, move water from the base of the shoot down the root axis into the tip. And while movement down the axis may be relatively easy because of the vascular development in that region, root tips are certainly not modified for rapid water transport. The newly produced cells don't have functional xylem, for example. But they do have phloem that moves all the way to the tip, obviously with at least one function of delivering sugars and other compounds to support root growth. And I'll return to that point in a second. So our conclusion at the time was that nodal root tips may be vulnerable to the development of low tissue water potentials when growing through very dry soil because of this characteristic. And so we asked whether this is a potential explanation of the difference between the two genotypes in our new data. And that certainly seems to be the case. This is showing you the apical 2 centimeter and then the 2 to 4 centimeter water potentials that we obtained in roots growing through a soil of either well-watered or -2 megapascals. And you can see that in both genotypes, the root axis just behind the tip actually has a very high water status. These roots are about 20 centimeters long, 15 to 20 centimeters long, deep in the soil. And so water can readily get from the base of the stem this far. It's not much different from the well-watered values. But to get into the tip where growth is occurring, as was mentioned actually, highlighted by Andrea in his presentation yesterday, the magnitude of the water potential gradient is going to depend upon the hydraulic conductance of the pathway. And in FR697, you see not much of a gradient in water potential required to pull the water into the tip to sustain root elongation, whereas a much steeper gradient is apparent in B73. Indicating that that hydraulic pathway just at the root apex through the growth zone appears to have a much lower hydraulic conductivity in B73 than FR697. Now, these are very new data. We don't have any mechanistic details of what the difference might be in hydraulic characteristics yet. But to try to link those observations with what I talked about in the first half of the presentation on the shortening of the growth zone that we saw many years ago in the primary root, I draw your attention to this paper from Wendy Silk that was published in 2009 in Plant Physiology, where Wendy was considering the path of water movement in maize nodal roots or other roots that have to grow through dry soil and are obtaining some, if not all, of their water supply downward from above. And she's considering here the impact of more apical phloem development on reducing the steepness of water potential gradients that develop to support growth. So on this key here, you can see that red indicates a lower water potential. So if there's no phloem delivery of water in this model, you get very steep gradients developing. But if the phloem, as the phloem is progressively more apically developing, that has the effect of diminishing the extent of these water potential gradients. And they speculate in this paper that adaptation to drought is predicted to involve more apical differentiation of phloem. And they speculate that that might be one reason why we often see this shortening of the growth zone, which in fact Wendy and I initially discovered in this 1988 paper that I showed you. So I emphasize in the first half of the talk our efforts to understand regulation of dimensions of the growth zone, and focusing today just specifically on our work on cell wall biochemistry. Whether that is or isn't related to the effect on hydraulics that I've shown you in nodal roots remains to be determined, but it's intriguing that understanding the regulation of the growth zone dimensions may be important in its own right, but also may have a secondary role in understanding the delivery of water to sustain growth in drying soil. Now, I'd just like to finish by moving from the lab model systems to the field. And this is really a pilot study. All of my work over the years has been in the very controlled lab systems I've described to you, without trying to tackle the more complex, much more complex field environment. And really understanding root growth physiology under drought in the field is pretty much a new frontier. And so, as part of this major new project we're undertaking, we're going to be doing that. But with my colleague Felix Ritchie, who developed this rainout shutter facility in Missouri, we are beginning to move our work from the lab, or continue in the lab, but also extend to the field. And so as an initial pilot study, because we don't know anything about what's going on in the field, and also to address whether we can get adequately reproducible data under drought conditions in the field, we had a graduate student, Hallie Thompson, who did this pilot metabolomic study in the 2013 and 14 field seasons. The experimental design is indicated here. We're harvesting the nodal root growth zone into two regions, the apical region A and 5 millimeters, and the 5 to 10 millimeter region B. I'll show you the growth kinematics in a second. And harvested the roots from 5 to 15 centimeters depth, focusing on node 2, which is the same node that we're focusing on in the lab. You can see in 2013, we only able to obtain relatively mild water stress with a root tip water potential of 0.49 megapascals, and we deliberately tried and succeeded to get more severe water stress levels in 2014. Now, as I mentioned, these data from the field show that this shortening of the growth zone that I've emphasized in the primary root in the lab does indeed occur in nodal roots under field conditions. And you can see this beginning to occur under the moderate stress in 2013, but quite obviously so under the more severe stress in 2014. This is the cortical cell length profile, showing you that in the well-watered roots, cells continue to expand until around 10 or 11 millimeters from the apex, but under water stress, cessation of cell expansion occurs at around 5 millimeters, very similar to the response we see in severely stressed primary roots in the lab. So we sampled these two halves of the root growth zone, sent the samples off to Metabolon Incorporated for a global untargeted metabolomic analysis. These overall results are shown here. This is just a statistical heat map for the 2 years, the 2 regions, showing the ratio of water stress to well watered for different metabolites. I don't want to go into details of that. I just want to emphasize what's in the Venn diagram. And this is for metabolites that were identified in both years, how much of those changes were reproducible across the years. And as you can see, 80% of the changes that we observed in 2013 were reproduced in 2014, which is pretty encouraging to proceed with more detailed studies in the field. Of course, there's a whole load more changes in 2014, presumably in association with the significantly greater stress that occurred that year. And I'm just going to finish just by showing you just one aspect of that complex data set to really link this field data with the lab data. And that is looking at the phenylpropanoid metabolism pathway leading to ferrolates and also on down to lignins. But I've emphasized to you in the primary root, we see ferrolate accumulation under water stress, particularly in the basal region of the growth zone where growth is inhibited. And that is certainly true in the field as well. In both years, this is 2013 A and B, and then 14 A and B. And the actual ratio data are shown here, with red meaning statistically significant, upregulated. And in the basal region in 2013, we saw a significant increase in both years. And in 2014, that was dramatically more enhanced and extending down into the apical region, which is consistent with that further shortening of the growth zone that we see under more severe stress. The other point I would just say from this is that another branch of phenylpropanoid metabolism, flavonoid biosynthesis, was one of the few changes in metabolites which were strongly and reproducibly downregulated in both years. And that's an interesting observation because of the known roles of flavonoid compounds in various processes, including ROS scavenging, modulation of auxin transport, and other processes. So we'll be pursuing that in more detail. So to summarize, I've tried to convey a little bit of the complexity of and coordination of mechanisms, emphasizing integration of morphogenic and metabolic events in regulating root growth under water deficits. The application of growth kinematics that we've used for many years in the primary root system, we're now extending, and it appears to be equally useful for understanding nodal root growth regulation. I focused a little bit on the role of ferulates in limiting expansion and indicated that modification of ferulate levels can lead to improved root growth both under well-watered and water stress conditions, and our initial results indicate very similar changes in cell wall metabolism in the field, so that suggests that ferulate chemistry might be important for root growth regulation in the field as well. And to return to where I started many years ago, our data rediscover the likely importance of root hydraulic properties in regulating nodal root elongation under very dry soil conditions, and bring out what appears to be a substantial genetic diversity in that response. And we'll be exploring that question in more detail as we move forward. Just like to thank a couple of grad students and postdocs that conducted this work, my colleague Felix Ritchie, that's allowing it to move to the field, and of course our funding sources. Thanks very much.
Thank you.
Graduate,
Thanks, Bob.
but